Hey everybody, today we are going to learn about biotechnology, studying and manipulating genomes. So genetic engineering is an amazing field, and it is the step where we study and manipulate DNA and RNA. Some techniques that are included are gel electrophoresis, PCR, which is polymerase chain reaction, DNA sequencing, and CRISPR. So gel electrophoresis, this is a great picture to draw. So you start with a DNA sample. It could be from a crime scene. It could be from a father who maybe doesn't know if he's the father. And what you do is you cut it with these enzymes called restriction enzymes. And the enzymes are specific to different sites. So for example, if I was to put my DNA with these restriction enzymes and someone else's DNA with the same restriction enzymes, it would cut our DNA at different locations because our sequence of DNA is different. So the restriction enzymes cleave or cut DNA into smaller fragments and sizes. And then what you do is you put them in the wells of a gel. So the gel is kind of like Jello, but scientific. And so you put the DNA in the wells, and the gel is floating in a buffer solution between a positive and negative electrode. So DNA is negative, and so it moves towards the positive. So when the electric current is passed through the chamber, so here's your wells, and the electricity is passed through, the DNA is going to move towards the positive end because it's negative. And if you think of smaller pieces going through a gel, the small pieces are going to move faster and they're going to be further up on the gel in whatever time you allow the gel to run. So that's gel electrophoresis, and then it separates based on the size. So you can compare two different individuals' DNA, or you can look at whatever different fragments for a disorder, or you could look for who's the daddy in a paternity test. So here's a couple examples of gel electrophoresis in different ways. So this would be, I know you can't see this, but this first one is DNA that was taken from a crime scene, and you can tell this one is the blood from the crime scene, and these are all the suspects. So if you're looking for whose blood was left at the crime scene, right, you would find the one that matches that. Can you guys figure it out? You should have picked number 3, right, because the bands all match. Now over here is what you would do for a paternity test. So here's Mary, here's Bob and Larry, and here's Mary's child, and she wants to know who's the daddy. So can you tell who the dad is? Remember, mom gives half and dad gives half. So you should have chosen Larry. So Mary gave the child this band. The next one came from Larry. If Bob was the daddy, then there would be a band right here. And then these two they got from Larry. So that's just a way that gel electrophoresis is used in real life. So the next thing you are going to learn about is what's called PCR. And we actually have a PCR machine in the classroom, and it stands for polymerase chain reaction. And it's just a fancy DNA copier. So like if you were to put, if you have a really small sample of DNA, you would put it in this PCR machine and it would amplify or make lots and lots of copies of the DNA that you need. And there's 3 main steps. First, you denature, and that's down here, and that's, splitting the 2 strands of DNA. So we heat it up, and then you put the primers on, and that's what's called annealing. So you put the primers on so DNA polymerase knows where to attach, and then you copy it and extend it. And then you would do this multiple times until you can get, millions of copies. And it's very automated. You just type in the length of time and how many cycles you want it to go through, and it just does it automatically. Another type of genetic technology and engineering is called bacterial transformation, which we're actually going to do in class. We are going to have some bacterial cells, E. coli, and we're going to get them to take up a foreign DNA that's going to make them antibiotic resistant and also glow in the dark. And so what we're going to do is we're going to make them what's called competent, which means that they want to take up the DNA, and that's by kind of heating them and cooling them and kind of getting them comfortable. And so here's your plasmid, here's your bacteria, and so the bacteria is going to take up the plasmid, and that plasmid can either be incorporated into the DNA of the bacteria, or it can just be a singular plasmid. And this bacterial transformation was how we amplified DNA before, right, before PCR. And what we would do is this would be the piece of DNA that we would want to copy. And so what we would do is we'd have the bacteria copy it because they can replicate themselves really quick. You can also use it to create medicine. So for example, insulin, you can take the gene for insulin and you can insert it into bacteria or a yeast cell, and then that yeast cell can make insulin instead of having to take it out of a person. And then also, genetically modified foods, we can do this. So DNA sequencing is important, and that is just like it sounds. It determines the sequence of DNA nucleotides, and we can use it in evolutionary biology to find out how closely related organisms are. We can use it in medicine and in forensics. And basically, nucleotides are flagged with fluorescent dye, and then they go through this gel, just very similar to the gel in gel electrophoresis. And each of these nucleotides are different colors. And so a laser is shown through the gel as the bases run through it, and they can detect what color it is, and they can sequence the DNA that way. Another interesting thing is how organisms are cloned. So Dolly was the first first animal that was cloned. And what you do for that, and you might want to stop and just draw the picture so you have an idea. So what you do is you take a donor cell from a sheep, right? Could be any cell because all of our cells are identical. And then you take an egg cell. And what you do with the egg cell is you take out the nucleus, and then you put them together, and they're fused together using a shock, right, to get them to combine. And then that fused cell becomes an embryo. And remember that nucleus came from this sheep over here. And since we don't have a nucleus in the egg, this nucleus has all the full set of chromosomes, and it develops into an embryo. And then that embryo is placed in the uterus of this mom, and then out pops a baby that's going to be identical to the donor. It's pretty interesting. They've cloned lots of things. They've cloned cats, they've cloned dogs. Just recently, last year, I think they announced they cloned a monkey, which is pretty crazy because that's very close to a human. So CRISPR is amazing, and it is going to revolutionize everything. And what CRISPR is it's a system that guides a protein called Cas9 to edit, cut, or replace genes. And so it's a three-step process. What happens is the scientists provide like an address, which is a section of DNA that they want to go in and manipulate. And then this Cas9 uses the address, which is an RNA molecule specific with different nucleotides for the section of DNA you want to go find. And it acts like a tracking device. And then it goes in there and either cuts out the section or cuts it out and rewrites by adding nucleotides. And it can go anywhere in your genes and manipulate anything as long as they have the right address or the right RNA. So it's pretty amazing. Something like sickle cell anemia, which is a base substitution, right? If you could go in there and just change it back to what it should be, then you could make it so people don't have sickle cell anemia. So very new technology in the last 10 or so years, and I'm sure will become even more prominent. And that's it. Oh, I don't know what I just hit, but have a great day. I am going to put 2 videos in the comments. So you need to go ahead and watch those videos in the comments under this, and hope you learned something about biotechnology.